I'm Norman Maitland. I'm a Professor of Molecular Biology at the University of York, where I'm also Director of the Cancer Research Unit. I want to understand both how prostate cancer develops, but also the best way to treat it. And in doing this, we want to study samples from real prostate cancer patients. Now, if you think about it, most people who are studying cancer work on cell lines. And these have been around in the lab for something like 40 years and have given a lot of useful information. But compared to life 40 years ago, life and cancer right now is very, very different. So we think that by taking this approach, we're studying a real cancer which affects real patients under today's circumstances and therefore is much more relevant to the development of treatments. Our approach to prostate cancer is very different. We've gone straight to analysing the complex mixture that's present in a real human cancer. Not just the tumour cells, but supporting cells, cells from the immune system, the blood supply to the cancer as well. To study it, perhaps perversely, we break the tumour apart, but then we study the individual cells and then we put them all back together again. By doing this, you get much, much deeper insights into what's going on in the cancer. But more importantly than that, we're getting very different results from the ones where a scientist maybe grinds up a whole tumour mixture, which is dominated by the major population there, or when he studies just the one population you get with an individual cell line. Our approach to studying cancers means we can pick out each of the individual populations within the cancer. What we really wanted to know was what a cancer needs to be able to establish itself, to be able to spread, but also what's there when we treat cells with drugs or with radiotherapy. What we found was there was a small proportion of very unusual cells which looked like stem cells within every cancer population we looked at. And these were remarkably homogeneous between different patients. And we called them cancer stem cells. Now, most people would say they're stem-like cells, but they form a root at the base of the cancer from which all the other cancer cells can be produced. They are present in about 1 in 1,000 cells within a cancer mass. And if you think of how difficult that is to see 1 in 1,000, then they are very difficult to study. So rather than find clever ways to make lots of them, we decided to shrink all the assays down so that we have miniaturized assays which include the stem cells. And since 2005, when the laboratory first reported this, we've developed these methods to study the cells. So we know what genes they express, we know lots of their basic biological properties, but still we had to find a way to study them together with all the other cells, picking out 1 in 1,000 cells. And that needs new forms of microscopy. So the Phasefocus system, which is also known as the LiveSight, allows us to look at individual populations of cells, all under different forms of treatment. And we can follow this treatment and the effects on the cells over a 5-day, perhaps even a 7-day period. We haven't taken it quite that long yet. But what you can see is the resistant cells at this time. Most of the cells succumb to chemotherapy very quickly. We follow the entire process, scanning regularly with time. So you can see the motility of the cells, particularly the stem cells, and you can see the effects that the drugs and radiotherapy are having on those cells. And suddenly your eyes are open to the fact that not all cancer cells respond in the same way. Now, if you do a snapshot, you get an average over 1,000, 10,000 cells. This way, every single cell in that population is a time point, is a data point. And we can follow it, and the software allows us to record all of this. The tychography system is not just a simple microscope. It relies absolutely on the analysis of the data. The Phasefocus software is absolutely essential. So looking forward into the future, what we really want to understand is why some men succumb to prostate cancer so quickly. Some of the population of what you could describe as a good cancer. These men can survive the rest of their lives with just monitoring of their disease. Other men develop from day one a dangerous, metastatic, lethal cancer type. and can die in as little as 2 years. The therapies help to suspend the end of life, but they don't cure anybody. And we don't understand, first of all, which drug should be given to which patient, but also how that drug, in many cases, is working or not working in the patients who aren't susceptible to the drug. If we can sort this out, it's not only better for the patients, because they will know that the treatment they're getting is going to work. It's also better for the health economics, because 70% of patients with advanced prostate cancer are given a drug which will not work, and we don't know why that's the case. So to improve the situation of men with advanced prostate cancer, something which is very dear to Prostate Cancer UK, who've given us a lot of money to study this. Also to myself, as a man now firmly in the at-risk group to develop prostate cancer.